Hi, my name is Simone Duran-Nyers, and today I'm going to be talking to you about my capstone project, which was centered around creating an in vitro blood-brain barrier. So, what is the blood-brain barrier? The blood-brain barrier is a separation between the brain's environment and the body's environment. And it basically keeps the brain in its own perfect environment that allows you to be alive and healthy. And the blood-brain barrier is characterized by endothelial cells, which line the blood vessels within your brain, forming tight junctions. These tight junctions are further supported by the feet of astrocytes in the brain, and together these two cells basically create a nearly impermeable barrier that only allows good stuff in and waste out. So, just giving you some examples of things that can cross the blood-brain barrier. Oxygen. Your brain is an oxygen hog. It loves oxygen. It can easily cross. Caffeine. My brain loves caffeine, and it can easily cross the blood-brain barrier. Hormones, right? The protein hormones, such as dopamine and endorphins, can cross the blood-brain barrier, as well as steroid hormones, such as estrogen and testosterone. And then ATP. Your brain loves ATP. It can easily cross the blood-brain barrier. So if we have this mechanism to protect ourselves and keep us essentially alive, why would we want to cross it? Well, when we have ailments of the brain, we want to send drugs in to treat them. In order to do that, we have to cross the blood-brain barrier. Some of these ailments could be brain tumors, hypoxic ischemic encephalopathy, which is characterized by low O2 in blood flow to the brain, causing neuronal deficits. And then anxiety and depression. So all of these things are brain ailments that we have to cross this blood-brain barrier in order to treat with drugs. So the main goal of my project was to create a clinically relevant in vitro blood-brain barrier with the hopes of testing potential central nervous system drugs on it. So how did I make a blood-brain barrier? Well, I cultured human brain endothelial cells. These are my cells. They're all nice and tight, forming those junctions. And then I cultured primary human astrocytes. These are my cells, and you can see their cute little feet ready to be supportive of those tight junctions. And I plated them together on a transwell insert. So this is a semi-permeable insert that goes into a cell culturing plate. And I put my astrocytes on, I waited 2 days, I put my endothelial cells on, and then those sat together for 7 days until I was ready to do more testing with them. And the top side represents the body, and the bottom side effectively represents the brain. So I can't just plate these cells and call it good. I have to actually confirm that I have a barrier. One way to do this is by transepithelial electrical resistance, or TEER, and I wanted that to be at or above 45 ohms per centimeter squared. That value was determined based off of previous literature. And this schematic just showcases how the TEER electrode works. One end goes to the bottom, one end stays in the top, and it measures the electrical resistance between the two compartments. I also used dye measurements, Evans blue and sodium fluorescent, which are standards in the neuroscience community. And how they work is that if you have a healthy blood-brain barrier, you're not going to present with these dyes, versus if there's a damaged blood-brain barrier, these dyes will be prevalent. So if I load dye onto this side and I have a strong barrier, not a lot should cross to that bottom side. So with this main goal, my hypothesis was if I plate these two cell types on that transwell, I should get that strong TEER as well as low dye flow through to the bottom. So I initiated my in vitro blood-brain barriers. I waited 7 days for them to form all those junctions. I did my TEER measurements and my dye measurements, but the results showed that I had improper barrier formation. So this graph will showcase the TEER with the proportion that data makes up of the whole data set on the right, as well as the frequency of that data point, how many times it occurs within the data set on the left, and then the TEER in ohms per centimeter squared on the bottom. So I did a one-sided t-test to see if my ohms, my TEER measures, were significantly greater than 45, and that yielded a p-value of 1, so the answer was no. They're actually significantly less than 45, but that's okay. You know, some strengths of TEER is that it is super sensitive, so it gives me super accurate measures and is ensuring that we only do experiments on actual in vitro blood-brain barriers. But a weakness with this sensitivity comes from me being a first-time user, right? Being perfect on the first time you do something is not usually attainable. So it's some learning curves for me. And then I wanted to make sure my plating was consistent between rows. I plated one row and then the next. So I did a t-test between those two rows just to make sure that I consistently plated cells. And there was no significant difference in the TEER value between the rows, which means I was consistent with my plating. Then looking at my Evans Blue results, I was actually really happy with these. My control showcases a higher amount of dye than my trans wells with cells, which is great. That showcases cells were in fact there and they were blocking some of that dye from going into the bottom. But when we look at our sodium fluorescent results, they are pretty convoluted. The cells in the control basically allowed the same amount of sodium fluorescent to the bottom, which is unfortunate. So the next steps are just polishing this blood-brain barrier establishment methodology and then testing Waixenicin A on an in vitro blood-brain barrier. So why should we care about Waixenicin A? It's a marine natural product and a potent TRPM7 channel inhibitor. It comes from Sarcothelia Edmondsoni. And TRPM7 perpetuates neuronal death in hypoxic ischemic brain injury, or HIBI. And HIBI is a major cause of death and chronic neuronal deficits in infants. 60% of preemies are affected by HIBI. So this was the whole motivation for my project. I want to optimize the BBB method so that YA's therapeutic potentials can be further investigated. Thank you. Any questions?